Hello and welcome to the Light-Sheet Chronicles. I'm Dr Elisabeth Kugler, a science communicator and your host of today's episode, Image Analysis in Light-Sheet Microscopy, Challenges and Possibilities. In this episode of the Light-Sheet Chronicles, I continue my conversation with Dr Kate McDole and Dr Jan Roden to explore data analysis and emerging advances in light-sheet microscopy. This episode will focus on sample and cell tracking, such as how to computationally follow thousands of cells during embryo development, or how to achieve real-time sample stabilization during imaging. We also address challenges such as registration, annotation, and benchmarking. With us today, we have two amazing guests. First, we have Dr. Kate McDole. She's a research group leader at the MRC Laboratory of Molecular Biology, or MRC LMB in Cambridge, and her lab explores the morphogenesis of early mouse embryos using a combination of advanced light-sheet microscopy, biology, computational methods, and biophysics. So welcome, Kate.
Nice to be here.
It's fabulous to have you on board. We really look forward to this conversation. And with us today is also Dr. Jan Roden. And Jan is a senior software architect at Bruker Luxembourg, the Light-Sheet division of Bruker. After studying physics in Germany, he has moved to the US for a postdoc in theoretical physics at UC Berkeley and Berkeley Lab. And since 2016, he is software architect at Bruker Luxembourg. So welcome, Jan.
Thank you. Thanks for having me.
It's fabulous to have you both. So maybe I can start off with asking you, Kate, because in your lab, you work with hundreds and thousands of cells, and obviously that can't be done manually. You have to kind of automate cell tracking, but cell tracking is very complex. So maybe you can introduce how you do cell tracking in the lab.
Yeah. So we primarily use cell tracking, and again, some of it depends on the particular biological question. But for the mouse embryo, our goal in being able to track every cell in the embryo is to not only know, okay, this cell came out of the primitive streak here and ended up, being a neuron. This cell came out and ended up being a cardiac cell, is to be able to actually trace lineages. So knowing where this cell divided and gave birth to these progeny, which gave birth to these progeny, and where did they end up? So do you have cell fate switches or cell fate bottlenecks? Where do lineages start to diverge? How similar are clones to each other? And then ultimately, can we take these lineages, if we can capture them over multiple days of development, and compare them to these transcriptomic data sets where we have all these RNA-seq data sets, these single-cell RNA-seq data sets that have predicted cell fate trajectories? Can we compare them to actual lineages in the embryo? And then can we do things like spatial transcriptomics? So where you image an embryo for a series of days, and then you take it out at the last time point, do some spatial transcriptomics on it, and then you have the molecular identity of those exact cells that you've tracked since, their origin out of the streak. And then you can also register that to other embryos that you've done spatial transcriptomics on at different time points. And then you can say, okay, well, there's not a one-to-one like in different embryos of the cell, like there is in C. elegans, because in mouse cells are not as, it's not a stereotypic development. You can say, okay, in this region of development where we know these cells came from, they're expressing, these genes at this point in time. And then you can see how that transcriptome profile evolves over time. And so that's really our goal with being able to do this cell tracking, is to be able to do this kind of lineaging. And then also with lineaging, you get divisions, kind of the orientation to divisions for free. If you know where the cells are dividing, then you also know kind of the direction that they're dividing in the embryo. So previously we've looked at division orientation as kind of a proxy for different tissue properties. So if a tissue is elongating, so the metaphase plate tends to orient along the axis of division. And so if cells are elongating in one direction, you'll have the metaphase plate align in a certain way. And so if we know where all the divisions are in embryo are happening, and maybe they're all random, and then at one time point, they suddenly all switch and start orienting in a certain direction, then mechanically something interesting is going on there, and then you can go and take a closer look. And so this is obviously very, very complex. A lot of it is difficult just due to the quality of the imaging. In some tissues, even on our microscope, they're just very, very, very scattering. And so it's hard to get complete tracks out of it. And there's a lot of, when you have issues with some of the signal to noise, if two cells get too close to each other, they can look like one cell. And so we're trying a number of different methods to train these neural networks to actually, predict the motion of the cells. And so that knows if two cells come close together, even if they look like they're fusing in an image, that those are two cells in the next time point, and to continue tracking those. Some of it is using some of the post-processing methods that are, again, mostly machine learning based to try and correct for some of the artifacts that are come from the imaging, or just in these regions where we just don't have the good signal to noise yet, then we're just not going to be able to track in those yet. But that's where things like mosaic reporters, where you just have a single cell labeled in that region, may be able to get us more information than just tracking every single cell in the embryo.
I could speak with you for hours about this. Thank you so much for this wonderful insight, because I think it's very interesting that, for example, cell proximity can be an issue, but you're using neural networks, but also really the whole intersample similarity, like are the tracks similar, are they dissimilar? There's really this whole scale of like data that you have to consider, which makes it super interesting from a data point, because obviously you need to really zoom in, but then also kind of zoom out a little bit. But I know, Jan, you use at Bruker a very different approach to sample tracking, because you don't actually look at single cells at the moment, but it's much more about tracking during the imaging, for example, where samples might drift or they grow out during the imaging. And so can you talk with us about why this is important and how you approach that?
Yeah, exactly. So this is a much less sophisticated, I would say, type of tracking than what Kate does, which sounds awesome, by the way. So we track more like the whole sample, right? So that when it starts drifting out of the field of view, that then the stages automatically brings it back into the middle of the field of view. And so the way we do this is we basically every time point during the acquisition, we run an image registration, and that detects whether the sample has moved and by how much. And then there's a configurable threshold where you can say, okay, if the sample has moved more than this and this, then start compensate the movement with the stage. And then so the software is sending the commands to the stage to move their sample back into the field of view. That's so much less sophisticated than what Kate was talking about with the cell.
But very important. Some samples like to run away. It's not even that they drift. They're afraid of light and they will swim away.
Oh, absolutely. And also just as examples of like cell growth, right? If we think about things like the lateral line in zebrafish, where it literally moves and you have to follow it over time, it's so important to have that tracking option. I had a lot of experiments that went wrong and my cells just grew out and you're just like, no, data loss. It's, I get it. It can be very frustrating. So it's good. And it's very, very important. And so Jan, with the tracking, I think what is also interesting, is it then like in XYZ or how does it work?
Yeah, exactly. So it's a 3D image registration, and it detects any sample drift or movement in any of these directions, and then can move the stage also in XYZ. And so it's not just for sample drift, but also some people look at growing plants or plant roots, and those can also grow out of the field of view over time, right? So if you, for example, want to keep imaging the tip of a growing root, then this also comes in handy, this type of tracking.
And you mentioned registration. And so I want to use the next few minutes maybe to talk about the importance of registration. So Jan, what type of registration do you use and what challenges do you face?
Yeah, I think, to me, from a data processing perspective, registration has always been the most challenging part of the pipeline. I don't know if you agree, Kate, but for me, it's such a important basis for everything that follows often, right? Because if you have a multi-view experiment, so for example, you have multiple objectives, or you have a rotation stage where you can have views from different rotation angles, and in the end you want to somehow fuse this data, or maybe even do a multi-view deconvolution of the data. And for all of this, the good, accurate image registration is the first crucial step, because otherwise you can't even do a multi-view deconvolution, right? If the data of the different views is not well aligned with each other, then it's the fusion most doesn't make sense, because then you won't be fusing basically the corresponding points in the sample, so to say. And for example, with these applications that Kate described, I can also imagine that if you want to keep tracking cells, for example, right, and now they are moving into part of the sample where the other view would actually be better. So then you need to somehow have a very good, accurate registration between the views, so that essentially the tracking can continue in the other view, so to say, if that makes sense. If when they are fused together, but if you have not sufficiently accurate registration, then this smoothly keeping track of the cells across different views is going to be difficult, right? And so then you also have on top of, so the samples can of course wobble because the stage is also moving, right? For example, if you do a tiled acquisition, whenever you move the stage, also depending on what kind of sample it is and how it is mounted, it can move a bit or wobble a bit. So this movement you need to compensate with the registration, but also optical distortions, right? Like especially if you image from orthogonal views, for example, in a multi-view microscope, there can be optical distortions. So you need often some kind of actually elastic registration that goes beyond like a global transform, but that can even align local elastic distortions well enough. And so, I think it's a very challenging part. And so what we are trying to do is we try to make this first step of the post-processing as good as possible over time, so that all the other steps of the pipeline that Kate described, all these sophisticated steps with tracking and AI and so on, that this can be based on a very good already, so to say, post-processed data that is already well aligned, right? So this is so that's why I think it's quite crucial to have a good registration as a first step of the pipeline often.
Yeah, I would agree with Jan, particularly when you have some samples, if they're sparsely labeled or there's not enough image content in the middle, it could be particularly challenging to fuse because you can't just rely on the coordinates of the sample or the stage to fuse it, especially if you're looking at fine structures. And in some cases, there are some data sets where we just can't register them. There's just not enough information, or if we're not tiling in the sample so large it's hollow in the middle, there's just no way to register it. And so then you just kind of have to cut your losses and take the best of each side, and then you get rid of the information in the middle where the image quality is not any good.
I love that. Thank you so much for bringing all of that up, Kate. That's really, really interesting. Especially, I think, the point about sparse data. And you mentioned before also that you do intersample registration and sometimes even mosaic labeling. So how do you approach it then if there's too sparse data? Do you fall back on like a manual registration with landmarks, or how do you approach it?
So in most cases, we also have a ubiquitous channel. So if we have one channel that's very mosaic, we have another channel that is ubiquitous. And so we can kind of use that as a reference. In some cases, though, where it is, if you just have a very sparse label, you're either going to have to do it manually somehow, or just accept the fact that you're not going to get both views fused together.
Love that. And do you ever use like fiduciary beads or markers?
We don't use beads in most of the samples simply because we can't use them in the mounting. So a lot of our things aren't mounted in gels or matrigel. So there's no way to really put beads in there.
Jan, thank you for all the really different aspects that you brought up. Maybe the same question back to you. Do you approach it with sometimes using fiduciary beads, or do you, for example, then have like fallback landmark registration?
Yeah, so we used to also have a special registration method for beads in the past, but we also noticed over time that, as Kate just mentioned, most users actually it's often not suitable or inconvenient to put beads into the sample. So so the alignment from a pure bead sample is not good enough, because as I described, the sample itself can wobble, so you would need beads inside the sample, which is often not really practical in many experiments. So we try actually to with our registration to be able to do without beads, and we've been able to make some significant progress in that direction recently. So I hope it's gonna be easier in the future. I mean, the beads-based registration methods, they are very different. They use very different algorithms, and it's much easier, but yeah, unfortunately, it's often not really in practice. It's not really so easy to put beads into your sample. So that's why we are trying without normally.
Yeah, I can agree. I've tried several kind of bead-based registrations and it was never quite successful. But I love what you mentioned quite a bit earlier, Jan, that you actually use a two-step approach of like first a global course registration and then a local fine-tuned registration, right? And I think that ties very nicely into what Kate mentioned with having a second channel that's where you have a ubiquitous signal, so to speak. And then you can use that as a kind of course registration and then use the secondary channel as fine registration. So do you think it's always necessary to have like a course and a fine registration, Jan? Or do you think sometimes you can get away with just a very quick and dirty registration?
Yeah, that depends very much on your application and what your following pipeline looks like, right? What do you want to do with the data in terms of analysis? And so often, for example, people who do cell counting or cell tracking, they need a quite accurate registration that is accurate to their single cell membrane often, because otherwise, cells are being miscounted, or like if you have doublings, then in the fused data, so that it's very application specific. So if you just want to have a rough correspondence between two channels, for example, you can of course get away with a more coarse-grained registration, but yeah, it really depends on the further steps of your pipeline, and also if you have, for example, other like AI tools in the pipeline, how well can they deal with these artifacts that come from inaccurate registration? Or if you, as I mentioned earlier, if you want to do multi-view deconvolution in order to sharpen your images, then yeah, normally a really accurate fine-grained registration is crucial. Otherwise, you lose more image quality through the inaccurate registration than you gain from the multi-view deconvolution often. So I think in those cases, it's very important to have a fine-grained registration. But if you just want to, let's say, roughly overlay two channels and just see very roughly which place corresponds to which place, then yeah, often like a rough global affine transform or something like this could be enough also.
So as we're running out of time, I know we wanted to spend a lot more time talking about data handling and storage, because obviously we could talk about light-sheet microscopy and data all day long. But to kind of really just wrap up, maybe Kate, you can talk with us. What is your kind of hope about data storage and the future for light-sheet microscopy in terms of how we handle data, how we store them, how we archive them?
Yes, because this is a big problem in light sheet microscopy, particularly as more and more labs have access to these technologies, which on the one hand is absolutely fantastic and it's great. But the long-term storage, the processing, the handling, and the dissemination of data sets when they're on the order of dozens of terabytes, even after they've been processed, is still a huge challenge. And a lot of labs, we still rely on sending hard drives back and forth. Over the pandemic, when everyone had time to do some data analysis, we sent 80 terabytes over Dropbox. This isn't sustainable as a model for every lab or for science to really be its most efficient going forward. And so having repositories, like some of the things EMBL or the IDR is doing, where you can put the data online and other people can access it. It's nice to see more of those initiatives going on. It would be great to have more of those. I think ultimately this is not just the long-term storage of mass data sets. It's not just a problem for Light-Sheet, but also, globally, we're running into this problem of how do you do cold storage, not only for energy efficiency, but also just the amount of data that we have. So for these Light-Sheet data sets, that are on the order of, a terabyte to 50 terabytes already, that can exceed the capacity of most labs. So a core facility can have one of these Light-Sheet microscopes and a single recording could spit out, 20, 30 terabytes. Where do you put that? And where do you put that long term? Specifically because most granting agencies require you to store data for 10 years, but try and get funding to repeatedly buy servers or cold storage or cloud storage for 10 years. And no one wants to fund that. So then what do you do with that data? Do you put it on tape? But then that makes it very hard to access and very hard to share with other people. At what point do you decide to delete that data? We're very judicious, I want to say, in how we store our data. So we take the Marie Kondo approach, which is you look at your data, and if it does not spark joy, if something happened with that data set, if something happened with that embryo, or you have a better one, you just delete it. If it's not perfect, you just delete it. You don't have the luxury of space to hold on to imperfect data sets when we run on order of 2 petabytes as it is. So it's a huge problem for the field is how do we store this data long term, and what do we do? What's the most efficient way of keeping it and storing it and disseminating it?
Oh, absolutely. Jan, what's your thoughts?
Yeah, I unfortunately also don't have a good solution for that either. Otherwise, I would have already suggested something. But it's definitely a big problem. And we often with our microscopes we try to provide additional data servers, but as Kate pointed out, you always run out of space eventually, right? And I guess another approach is to, instead of storing all the raw data, storing the already some like segmented data or so that is smaller, some data in some form of representation that is more compact. But then, of course, you have the problem that you never know what you want to do with the data in the future. And if you delete the raw data, you don't know, right? Maybe it could have been useful for some other kind of analysis or so that you hadn't foreseen in the past. So it's always this pickle that you are in. So I don't know a good answer to that either, of course.
Thank you so much for those insights. I think we spoke about loads of things today, like only keeping the good data, making sure we have like optimizing the acquisition. I think that's lots of like also AI work obviously happening, but also what we kind of spoke about at the very start about kind of keeping sparse data. We kind of remove the empty pixels, probably one of the main challenges to be solved at the moment. So thank you so much both for bringing that in as well. But unfortunately, we have to kind of start wrapping up. So I do have a very quick fire question, just very quick answer. What's your favorite part about image analysis, Kate?
For me, a lot of the things we look at, no one has ever seen before. So actually being able to look at these data sets and start going through and seeing a lot of these processes and things, structures, and even like the heart appear, no one has ever seen that before, certainly not at that resolution. And so that's always very cool. I will just sit there and play data back and forth and go through in 3D and look at things and say, oh, how are those endothelial cells coming together? And, ooh, look, you can see that. And, ooh, look, you can see that. And so we just seeing the whole picture finally all put together.
So what science is all about, discovery and curiosity. That sounds amazing. And for you, Jan, what's your favorite part of image analysis?
Yeah, I just love to see, people in the labs use what we've developed in terms of software and microscopes, and to see what it enables them to do and how creative they are in applying these things. And often, I love sitting next to someone using the software that I've developed, and they can actually use it faster and more efficiently than I can use it myself. So these are the moments where I'm like, okay, that's awesome to see it being used by someone this efficiently. And yeah, so those are the moments that I really love.
That's amazing. Thank you so much for sharing that. And I'm afraid that's all that we have time for today. So thank you both for joining in.
Thank you.
Thank you. Thanks for having me.
It was amazing. So thank you so much for your time and all the insights that you shared. Like I say, I should have known you during my PhD. It would have saved me a lot of time. And so today we have talked about biomedical image analysis and light-sheet fluorescence microscopy, including processing, handling and storage of big data. And so to our listeners, if you want to know more about biomedical image analysis for the Bruker Light-Sheet Microscopy division, make sure to check out our Lux Bundle Tech Note, which is linked below. And so to our listeners, many thanks for tuning in.
This podcast was sponsored by Bruker Luxendo GmbH, the Light-Sheet division of Bruker's Fluorescence Microscopy division. Bruker is a manufacturer of scientific instruments for life sciences, materials research, and industrial or applied analysis. If you'd like to learn more, please visit www.bruker.com/light-sheet. This podcast was produced by Keep It Science.